Hi guys, welcome to Science Tech for Fun. This is Mr. Fasten here, and today I will be talking about, that will be a long one, biology practical number 11, production of dilution series of glucose and a use of colorimetric techniques to produce a calibration curve and identify concentration of glucose in unknown urine samples. Oof, that was very right. For this practical, this is all the materials involved. You can see here the materials, and as long as I start to do, I will explain what I'm doing and what I'm using. Right, for this first part, the first thing I need to do is look after myself. As I always stress, PPE, personal protection equipment. For this practical, is standard spec safes. If I'm using chemicals, gloves, lab coat. Also, I'm using a water bath with very hot water. So gloves and lab coat is important to protect my skin as well. Okay. First thing I need to do is to set my water bath. The method says I need to set in about 90 degrees. This will take long to reach the temperature. So the first thing I need to do is starting water bath and let it heat it up itself. Okay. Second part is I need 9 test tubes, 2 small beakers, my glucose standard solution, that's 10 nanograms per liter. I need my Benedict quantitative solution and distilled water. So what I'm gonna do is I will label my test tubes. I will label from 0, 2, 4, 6, 8, 10, and my 3 samples of urine as Tom, Dick, and Harry. Okay, so these 3 will be my unknown samples of urine, and these 6 test tubes are for the curve. Okay, I also need 2 beakers. The 2 beakers I need to label with water and Benedict, so I will place it here. And then I have my original sample glucose, my Benedict, and my water. Okay, so after I labeled everything, I will go for the solution, how to do it. For the calibration, that's my first part, I will need to mix water and glucose. The concentrations, the ratio between water and glucose should be calculated by you. I will show a little table how to do it, but you need to calculate yourself first. The final volume of solution between water and glucose is 2 ml, so 2 ml in each of these test tubes. Okay, so take a look in the table now. Right, so let's start with the solutions. Okay, so it's better if you do all water first and then all glucose later, or the other way, all glucose once and then water. Okay, so I will start with water. So for my 0, I will need 2 ml of water and 0 ml of glucose. So here I have a 2 ml pipette, and I will suck 2 ml. So now I'm sucking 2 ml of water. When reach the meniscus, I just transfer for 0 here, and I will displace the liquid. When it's almost at the end of my tube, I can squeeze off, rotating this part here to expel the rest of the liquid. And I will repeat the procedure for everything. After I'm done with water, I remove my pipette, and I'm gonna place a second one. The second one will be just for glucose. So I will repeat again. So the 0 one, no glucose. The number 2, it was 1.6 of water, and I need to top up for 2. So it's 0.4 of glucose. So I will see here. So 0.4. So we start here. So 1.8, 1.6. So this is 0.4 ml of solution. So I will open my bottle, put it here inside, and I will collect 0.4 ml of glucose. Then I will transfer for my tube. Press the release button. When it's almost finishing, I rotate the round off to expel the rest. And I will repeat for the rest of the samples. After you produce your solution, it's a good idea to check the levels, okay? So you can see the levels are all the same. So take a look, the levels is about the same. So you guarantee that you have 2 ml in each test tube, so you didn't mix up. So after you guaranteed you have the 2 ml of your samples for the calibration, you will transfer the 2 ml samples of Tom, Dick, and Harry for the right test tube, and then you're gonna add 2 ml of Benedict solution quantitative in the 9 test tubes. If you don't have pipettes enough in your school, you can just, after using the sample, you can just clean up with distilled water and dispose, okay? So you just suck water, release the water, so you guarantee you can use again, and the sample will be removed from inside. So after all the volumes are checked and displaced, give a small visual check and see if the levels are correct, okay? And then everything ready, we will transfer the 9 tubes for the water bath. We're gonna keep it for about 4 to 5 minutes. After that, we'll remove from the water bath, let it cool down a bit, and then we go for the colorimetric technique, okay? Okay, so after 5 minutes in the water bath, I remove it and kept it outside for a couple of minutes to cool down as well, okay? So this is my initial results. You can see a grade of colors here, okay? So it's visually possible to see there is difference in grades of milky white color, okay? So what I will do now is I will first start my machine, my colorimeter, and I will calibrate with pure water. So the method says to calibrate the machine, calibrate for the curve with the 0 initially, just water and Benedict. But before that, you need to calibrate your machine with pure water. Otherwise, the machine is not gonna read properly. So then, I take my cuvette and I will add water. And the cuvette fits about 4 ml of liquid. So I have liquid enough to just pour here inside. So I will put here. Oh, before that, let me show you how to assemble the machine, okay? So here in this model machine, the Mystic, you just have the on, okay? And then you have AT. A stands for absorbance, T stands for transmission. We need absorbance. So I will change here when it shows A here on the side, okay? So when it goes for A, and then I have this button here say RGB. RGB stands for R red, G green, B blue. I have to choose the color that goes most opposite to the sample I have. So the most opposite to the sample I have is red. So I will click here until on this side shows R. So now it's showing R here, okay? So now I will put back here in the table. I will place my cuvette with water inside, close it, and I will press Cal for calibration. Right after calibration, this is gonna zero everything, and then I will place my first sample, that's the 0 glucose, and then I will go for 0, 2, 4, 6, 8, 10. I will write my results, and after that I will do my Tom, Dick, and Harry, and I will compare the results between the three with the curve that I did, and then I will be able to find my unknown results for my urine sample. Okay, so I will show you how to do the first. So everything is ready here, so that it is put here inside, cover, and then I press cup. So now it's showing 0, 0, 0, 0. So I open this up, take this off, and to be easy for me to know what I use and what not, I will keep my empty ones in the bottom of my rack, and every time I use, I put it in the front. So I will start with my 0, transfer here. Look, fits exactly the right amount. And then I will put here to the front. Place inside my cuvette, reader, and close. So now is reading 0.895. So before you transfer the samples for the cuvettes, you will see that the bottom of the test tube will have like a small amount of solid. You have to shake it to make the solids goes up, because this will change your results. Otherwise you might have mistaken results. So always mix it before you pour in your cuvette, okay? Another thing, after you place it inside the machine, let it be for a bit to stabilize, okay? If you see the numbers going up and down, this means the solution is not stable yet. So let it be for a couple of seconds and then you take the reading. And what might happen as well, for saving power, the machine will turn off. But even if the machine turns off, all the calibration will still save in the memory. So you just need to press the on again, and this will come back for your final calibration results. Okay, so this is going to be saved here. So even if this goes off, just press on again, and then you carry on with your readings. Okay, after reading, I can open up, remove my cuvette, put it here in the front, take another cuvette, and go for the next sample. So my next sample will be 2. Always check. Put it here inside. Don't forget to close the lid. And then I have my reading. So my next reading is 0.286. And then you carry on with your sample. Thank you for watching this video. If you like it, please give us a thumbs up. Please consider to subscribe. Give us a comment and see you next time.